Okay, we're continuing with the process of drug development. I put some stuff up there already, so kind of like a PowerPoint. So somebody at the company reads some sort of academic research, or somebody in their pure research area goes, this is what we think is working in these cells during this process. Well, the first thing they've got to do is try to test to see whether it actually works that way. Is that academic research is based on publication, and promotions and holding on to your job in a university very often depends on getting things published. So sometimes people rush it through. They don't always do the numbers the way that they should. They skew things in order to make them look better, and the peer review process should work better than it does, but sometimes things slip through the peer review process. So the first thing you got to do is make sure that what they think is going on is actually going on. So you confirm, you run your own in-house tests, and a decent amount of the time, your own researchers come back and go, well, I don't know what they're talking about. We can't find this, we can't get it to work this way, and so you're on to something else. You also have some fairly sophisticated computers where you can put things into the computer models to see and have them manipulate things in a simulation environment to see whether what you think you might be able to get to work will work as far as simulation is concerned. If you've got cell cultures for things like cancer and certain types of diseases that are very cellular, where you can actually keep the cells alive in a dish in the lab, you can take your patented molecule and expose the cell cultures to it, and then see how it interacts in cells that are completely isolated on a nutrient dish, so not involved in an overall connected environment like a person. But it gives you some idea of whether something will work in living cells, which again is a step up from the complexity. Computer model only knows what you know. It can put it together in some pretty sophisticated ways, but its limitations. The cells are much more complicated. We still don't know everything that's going on in a cell all the time. So the cell cultures look okay. Then you go to animal models, if you can. You need, if you're gonna develop a drug for a particular condition, you need an animal that has something equivalent to that condition. It may not be exactly the same condition, but it's close. It turns out for coronavirus, hamsters, mice don't tend to do, they'll support the virus, but it doesn't make them sick, and they seem to fight it off pretty quickly. Hamsters actually mimic the course of COVID-19 disease, so they have been used as an animal model, a couple of other animals as well. And in some cases, people have fiddled with the genetics of strains of mice to get them to mimic a condition that they wouldn't normally get. Like there's a mouse strain for multiple sclerosis. Now, mice don't normally get multiple sclerosis, but you can produce a strain that has what looks very much like multiple sclerosis. Now, it doesn't work exactly the same way after you fiddled with it to get it to show up. That's always the question. And does any of this stuff work in mice exactly the way it does in people is another huge question, or whatever animal model you're using. But it's a whole living thing. And so it's a much better test for whether what you're trying to develop is going to do what you hope it to do than the computer models or the cell cultures that you've used before this. And you have to do these things and write them all up and go to the Food and Drug Administration and go, here's our results. And somebody looks over them who has background in this, right, peer review. There's a lot of talk about how there's kind of a bounce back and forth between pharmaceutical companies and the Food and Drug Administration. A lot of people in Food and Drug Administration came there from pharmaceutical companies. And people go, well, then they're biased about these companies. They may be biased toward the company they came from. Maybe they're biased against the company they came from. Why are we doing this? Well, you need people who understand how all this stuff works. And it's a fairly small population of researchers that you have to dip into to produce Food and Drug Administration reviewers. So yeah, it's not a perfect system, but you want people with experience, you want people who understand how it works, so you're kind of stuck with the way that the system works. While you are working through this, this is also part of the animal models that's useful. So you've got this patented molecule. It has a target in a cell that's associated with some condition. How do we get this molecule from where we're making it in the lab into that particular cell in a whole body, in a form that it will still do what we want it to do? It needs to be deliverable. So just having the molecule and just finding out that it works in the computer and even works in a dish of cell cultures doesn't mean you've got something that will come in through the digestive system properly, will go through the liver without being altered. Or sometimes you have to factor that in, put a molecule in that the liver will actually change into the molecule that you want. Will it produce allergies fairly easily? If you have a chemical that you are going to throw at a chronic condition, so you're going to give it to people over and over and over, if they easily develop allergies to it, that's not going to be really deliverable over the long term. And not too surprisingly, it's got to be something that once it's approved, you can actually make it in decent amounts that you can actually sell it and make some money on it. So the FDA says, okay, we like what we see on paper, you can now test in humans. This is when you're trying to find out a few things. Things that work in mice don't always work in humans. Things that are not toxic to mice are sometimes toxic in humans. Does it actually have a significant effect on the condition you're looking to get? All of those things have to be tested in humans, and it's done in steps. When you look at it, in very careful steps. And I think I've mentioned before, once you're here, only about 1 in every 10 drugs will make it through. A little bit more for vaccines, but still the odds are against you actually getting to the end and getting approval for sale. For phase 1, you have a small group of paid volunteers, and they are not sick with the condition that you want to test it on. They are perfectly healthy people. Why? We want to know if this thing is potentially dangerous. So we take a small group, we don't want to endanger a big group, and we test, depending on what you're looking for, maybe different doses, but we're looking for toxic or significantly bad side effects in our small phase 1 group. And if it shows toxicity or it shows some major potentially damaging side effect, it's never going to make it out of phase 1. Because again, you have to do it, you have to gather your results, they have to go to the Food and Drug Administration, they have to look at it, and then you can see why this takes a while. And then they come back to you and go, no, actually, if your results are really bad, you never even send them in. But if your results are kind of on the fence or good, you send them in, takes a while, they come back, go, okay, phase 2. Phase 2, typically a larger group, but not a huge group. And these are actually people with the condition that you're targeting. Maybe you run a control group with this, maybe you don't. What you're trying to find out here is, does it look like it's going to work? and we're going to keep looking for side effects. So we've got a slightly bigger group, and they actually are sick. So a side effect that didn't show up in healthy people may show up in sick people with this particular condition. Here you run into a bit of a problem. You got to get people into the group to test. But it's not a big group. But you have to pick people. So you make a list of, we want people with the condition and we want this, 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 and this. In a perfect world, you want people that are sick with just the illness you're targeting and not a bunch of other illnesses. You want people that are not taking a bunch of other pharmaceuticals or drugs of any kind if you can help it. So you're going to select a group that isn't going to exactly match the wide group of phase 3 or the wide group of the world, is that you may be selecting people who are already leaning toward doing well. They might have gotten over this, whatever you're testing, they might have gotten over this on their own because they're pretty healthy. They don't have other conditions. They're not taking a bunch of other chemicals. But it makes sense, but it still biases your results. So very often, if phase 2 results are released, oh, the drug's working really, really great. And they'll do this in order to get investors to bump up their stock prices. You will get a lot of hype. And sometimes this happens where you hear, oh, this happened, this was really, really successful. And it's like, 10 years later, you go, whatever that happened to that? Well, it does a couple of things. There is something they call regression to the mean, where your average effect here is very good. But for a very large group where you do have these other issues, the real average in a real group is down here. And when you go to your bigger tests, this really good effect is going to regress to the actual average that's a mean. And maybe that's not sufficient to get you approved. But that is phase 3. Phase 3 is a big group. Is that with the coronavirus, they've been testing thousands and thousands of people with these vaccines. It's a controlled experiment. You have your group split up, matched to a large extent. Okay, we got a 65-year-old white male with these underlying comorbidities in our test group. We wanted a white male of that age with, as close as we can get with the same comorbidities, other diseases. And so you're not just making 2 groups. You're trying to match them for things that could be significant, like age, like gender, like ethnic groups, like other conditions that you're going to have because it's a big group, and you're not going to be able to select just the very healthiest people. But you want your results to be comparable. So you want these 2 groups to be similar to one another. Wow, that was a weird one. It is flu season. I just got my flu shot though, so. Anyway, you want your 2 groups to match as closely as you can get them to match, based on things you think are important. Is that, you don't match them for height unless you're doing a test on like growth hormone or something. You just match them on things that you think are going to be important. And now you're looking for, you have to decide, what are we looking for? What are we measuring? What effect are we expecting to get? Okay, are we going to measure 1 effect? Are we going to measure 2 effects? Are we going to measure 3 effects? Just to see an example, because it's in the world, with the coronavirus vaccine, they are looking for an antibody response, but that isn't, that's a good response for initial immunity. But there's also a couple of different immune cells that you can track. That's expensive. That's slow to produce the results. But it's a much better measurement of potential long-term immunity from a vaccine. And so they have, not only do they have to decide to do that, but at some point somebody at the FDA goes, no, antibodies isn't going to be enough. You have to do this. And what they decided in the coronavirus vaccine tests is that it doesn't have to produce an absolute immunity where you don't harbor the virus to the point where it's measurable, which for a lot of our immunities, that's true. You're not gonna, like if you're immune to the measles, you're not gonna support measles so that you would test positive afterward. Here, we're okay if people pick up the coronavirus and test positive. We want their symptoms to be pretty much nothing. So they're looking for a reduction in illness more than an absolute immunity. What you measure. So you do your big group. Takes a while. You need participants. If you've got thousands and thousands of patients, you're not doing this in your own pharmaceutical labs. You are conscripting doctors and medical systems. The coronavirus vaccine tests are happening all over the world. And they have to run the test the way that you need the test to be run, which means you need to have people out there observing them. You need to pay them. You need facilities that you're going to pay for. The prices get cranked up and up and up. This is very often hundreds of millions of dollars in a phase 3 test. One other thing for this, what if your drug is for a disease that's a very, a big deal. It's a deadly, it's nasty, it gets a lot of attention, but there's just not that many people that have it. There's not thousands and thousands of people you could pull into a trial. Well, you do what you can do, is that maybe you don't even run a control group. Maybe 100 people is all you can get, and rather than split that 50 and 50, you say, okay, we will compare the results here against the results of people who we know the course of this drug goes in people that haven't been treated. What those are called are orphan drugs or orphan conditions. And in the last 20 years, there's been a lot of government subsidy programs to support this research, because if there's not that many people with the disease, it's still going to be expensive to test, and you're not going to have a big population to make your money back on. So the government underwrites the development of this stuff. And then the charges still are fairly large, so that's a debatable subject where, okay, if you got a bunch of money to develop this, then the government ought to be able to set your prices on this stuff when you go to sell it. Sounds reasonable. The drug companies go, no, look at our costs here. We're not making money on this thing. Of course, there's their books and then there's the reality. You don't really know. If you're watching television and you see them selling drugs, turns out they didn't used to be able to do that. You used not to be able to market drugs to the public through commercials, because the idea that you could push people with no real training to go to their doctor and go, I want this drug, I saw it on the TV. They decided that was a bad model for pharmaceuticals. And then somewhere, I think in the 1970s or 1980s, the drug companies have a lot of money invested in our government. And so they have a lot of push to get things finagled, and they got the restriction lifted. So now you watch late night TV, if you're watching, any kind of channel that old people tend to watch, there's a lot of drug ads. And they give you the list of side effects. Side effects are worked out in phase 3. So big group, you're gonna see side effects. The side effects come in the order of how often did they happen. From the most common side effects in the group, and you don't know how many of the group it affected. Did it affect 5%? Did it affect 15%? Did it affect 40%? But it's the top number. And then they get rarer and rarer and rarer and rarer. I once had a reaction to a stomach drug where I went to the pharmacist and he went, no, that doesn't happen. And he looked down the list. They didn't give us the list back then. He looked down the list and said, oh, wow, that's second from the bottom, which meant it was an extremely rare side effect, but it was recorded in the data that the company produced. So your phase 3 goes great, and you hit your benchmarks that you're supposed to hit, and you send all of this data off to the government, and sometimes you go, well, overall it didn't do that great, but look at these demographic groups. It was really good for African Americans, or it was really good for people under the age of 30, or people over the age of 30. And anything you can do to try to convince the government to allow you to market the drug. Because phase 3 for most drugs is the last stage. And once they are approved, they can sell it. And the government doesn't set the prices, the company sets the prices. And pharmaceutical companies make pretty hefty profits. Is that, yeah, they're making back a lot of money that allows them to keep developing other drugs, but they're making a decent amount of money too. Now, phase 4 is only official for pediatric drugs that are going to be given to kids, where once they're on the market, they do require doctors to report back their, I don't know exactly what level of report backs, side effects for sure. I don't think you have to report back every time you dispense one, but anything weird that happens, they want to know officially. In most other drugs is that phase 4 is unofficial. And a lot of times phase 4 works where doctors talk to each other. They get together at conventions and they go, you know, have you tried this new thing? And somebody goes, yeah, I tried it. You know, one of my patients had this weird result that wasn't on the list of side effects. And, you know, you hear a couple of other doctors in the room go, oh, yeah, that happened to one of mine too. Somebody goes, well, it happened to 3 of mine. And so a number of years ago, a drug that was an arthritis drug that happened to be producing strokes in a fairly low number of people. But it was this getting together of doctors that then they went to the FDA and went, you guys need to look into this. This is looking like it's more of a potential side effect than they thought it was. And it turns out that they had strokes happen in the original phase 3 tests. But when they, at least this is a drug company, they said, we didn't think it was associated with the drug, is that all of them had these other risk factors that we thought were much more, you know, likely to produce the stroke, whether they were or weren't on the drug. So we didn't list it as a side effect, and the FDA went, you know, whack upside the head, pull it off the market, retest it. It's back on the market. Possible strokes is now on the side effects list. I don't know how far down it is, but no. But they also got fined a decent amount of money. This is called clinical testing. When things are advertised as being clinically tested, a lot of times they have not. And if they lie about it, it's not an FDA issue, it's an advertising issue. It's the Department of Commerce has to, you know, you can complain to the Department of Commerce to go, you can't use that in your advertisements. Or it's something that they did test in phase 1. It didn't get past phase 1, but they did test it in phase 1 so they can go, this has been clinically tested. A lot of this stuff that they sell on TV, people have tested this stuff and it has failed. Is that you don't get results that are any better than the placebo group with your test group. But if it's technically food, if it's a chemical that's in stuff that people eat, it doesn't have to pass these tests. It can be marketed, it just can't be marketed as a pharmaceutical. And there's some rules about that, have to do with advertising, have to do with what you can claim. But there's a tremendous amount of stuff out where they make all these huge claims. And if you look at most of the claims and you know anything about how any of this stuff works, you realize, okay, this is bogus, because, you know, it affects all these different conditions. Well, no, it doesn't. Oh, there's no side effects. Well, if there's no side effects, there's no effects. You get side effects because these things do mess with your chemistry, and you're going to get side effects from things that mess with your chemistry. As I mentioned when I started this, medical devices don't have this same level of testing that you need to go through in order to get approval. Surgical techniques don't. Psychiatric techniques don't. So a lot of what's called alternative medicine is only alternative because it has failed to pass testing. Is that there was a big government program over the last 20 years to test a bunch of this stuff. There was a senator who was a big believer in it. And so they put a lot of money into it. And then the senator was complaining like 12, 15 years in that I don't want to fund you anymore. You keep finding that the stuff doesn't work. And, you know, so that's the way that works. Okay, that is it for the entire course. That was the end of my notes. Hopefully you've made it this far. I always ask the phase question, so I hope you made it this far. And bye.